everyone, my name is Sophia Dominguez. Today I wanted to talk about the use of antibodies in research. Antibodies are a powerful tool which allows scientists to redirect proteins to different locations or analyze proteins of interest by tracking them. Before this class, I had always known antibodies were used for important experiments such as Western blots or immunoprecipitation, but had never understood their role. Antibodies are large Y-shaped protein complexes produced by plasma cells. There are 2 heavy chains and 2 light chains. They are used by the immune system to identify and neutralize foreign objects such as bacteria and viruses. The antibody recognizes a unique part of the foreign target called the antigen. The tip of the Y of an antibody is specific for one particular epitope of an antigen. With this mechanism, an antibody can tag a microbe or infected cell for attack or neutralization. The Fc region of the antibody is important because it is a constant region, which is important for antibody recognition and indirect immunofluorescence. There are 2 different types of antibodies: primary, which goes first and binds to the antigen, and secondary, which recognizes the constant region of the primary antibody. Antibodies can be conjugated to fluorescent proteins so that they are visible under a microscope. Monoclonal antibodies contain one specific antigen binding site, while polyclonal antibodies contain many specific binding sites. To make polyclonal antibodies, you first start by injecting the animal with whatever you want it to formulate an immune response to, which is a purified protein of interest, or an antigen. This will normally consist of multiple injections in order to ensure the species is getting an immune response. You will then draw blood from the species and get the serum through letting the blood clot, spinning down the clot, and collecting the supernatant. The supernatant will have all of the antibodies that the species has accumulated throughout the course of its life. You then need to elute the specific antibody through using a column. This involves the use of beads coated with your antigen. After putting the serum on top of your beads, discard the flow-through as these are not your antibodies of interest. Elute the antibodies of interest using a different salt concentration. The construction of monoclonal antibodies consists of making a fusion between the spleen cells of a mouse challenged with antigen and a myeloma cell, which is a cancer cell line from B cells. You get DNA from both in the fused cells, resulting in hybridomas. Each hybridoma will be secreting a different antibody. You can then figure out which hybridomas are making the antibody you want and expand that cell line. Monoclonal antibodies are an inexhaustible source that you can continue to grow. This concludes my brief presentation about antibodies. Thank you so much for listening. Bye.